So, welcome to the 6th episode of the S1E6 podcast. And today we have a special guest, Dr. Leonidas Chouliaras, a consultant of old age psychiatry, assistant professor in the University of Cambridge, and a member of the Essex Partnership NHS Foundation. We know that you're involved with various areas of research, including epigenetics when it comes to neurodegenerative diseases, but you've put out some articles regarding biomarkers for detection of these diseases. So we just want to start with an introduction about yourself. So can you describe your current title and your current work, and who are you, of course?
Yeah, so thank you both for inviting me. I'm delighted to be part of this podcast, and I hope our listeners enjoy that. So I'm a clinical academic old age psychiatrist. I spent about half of my time doing clinical work. I work in an NHS community old age psychiatry service looking after people mostly with dementia. And I also work at the University of Cambridge where I do research that's around improving our understanding of neurodegenerative diseases, focusing on genetics and epigenetics, and also been doing some work on fluid biomarkers, trying to improve the diagnosis, particularly the differential diagnosis between Alzheimer's disease and dementia with Lewy bodies. And we do that through various different ways. We try to look at fluid biomarkers of neurodegeneration, mostly in blood, but we also try to find new biomarkers focusing on the immune system, on epigenetics. And lately, a new project we're about to start is we're about to do a study looking at multi-scale proteomics to find new ways of improving our diagnosis of dementia with Lewy bodies. I wanted to say for the listeners perhaps who are not familiar, dementia with Lewy bodies is probably the most common type of dementia you've never heard of. It's the second most common type of dementia after Alzheimer's disease. It is very, very much undiagnosed. Research from post-mortem studies show that it's the pathology associated with the condition, Lewy bodies, alpha-synuclein deposits, is present in about 20% of post-mortem brain collections of people with dementia, while clinically the statistics suggest we diagnose it in under 5% of cases. So a large number of cases actually are misdiagnosed, and that's why it's important to do more research in biomarkers so we can improve the diagnosis of this condition.
So we've talked about biomarkers in this podcast before, but it would be nice if you gave us your definition of what a biomarker is.
Well, I guess it's a very good question. For me as a psychiatrist or as a neuroscientist, a biomarker is a marker of what is happening in the brain, I guess. When we talk about peripheral fluid biomarkers, it's something that can give us an indication about what is going on in this person's brain, what type of pathology is being deposited, what other problems are going on. And actually, the last few years, this has been revolutionized. Yeah. When we talk about Alzheimer's disease, we know people with Alzheimer's disease get amyloid beta plaques, and this leads to phosphorylated tau, which then leads to tangles. And there are now very good fluid biomarkers in CSF, in cerebrospinal fluid. We use them as diagnostic tests, and they are coming in blood. Markers of the pathology in the brain that we can measure in blood very accurately in very small quantities. And phosphorylated tau seems to be the one that is the most accurate fluid biomarker when it comes to detecting Alzheimer's disease pathology.
Mm-hmm. We read your study about the differential levels of plasma biomarkers, and we just wanted a clarification. We are university students, so we are learning nomenclature at this time, and we're wondering, what is a longitudinal study?
Yeah, I mean, it's a nice point that you say you're medical students and you're still learning. I think the learning never stops. I'm many years away from medical school, and I still learn every day, more now than what I did in medical school. So yeah, that's an ongoing process in medicine and research. I guess when we talk about longitudinal studies is when we see people in cohorts over several years and have an idea of what happens over time. Obviously, we see people at one time point, but people evolve, people change, people get worse or get better. In people with dementia, they mostly get better over time. And it's important to understand how quickly they get worse, if the progression of certain diseases is worse than others, because that helps us to know both at the point when we diagnose the people. I often work in memory clinics. I tell people you have this terrible condition or the other. And one of the common questions I get is, how many years do I have? What's going to happen next? And through research, we can understand that. But also if we try new treatments in clinical trials, knowing the natural progression of a disease, how quickly someone will get worse, will help us then know if a new treatment is working, if it's slowing it down, if it's stopping it. And I guess that's why we do longitudinal studies. So seeing people at multiple time points, usually we try to see people in several studies depending on the protocol every year. But some studies see people every 6 months or longer intervals. All depends on the design of the study. But that gives us an idea of the trajectory of the condition that each person has. And it varies a lot. One person with Alzheimer's or another type of dementia may deteriorate very quickly, and another person may be more stable for a bit longer.
I feel like a good follow-up would actually be kind of why you feel like earlier detection would be beneficial. So from like a clinical standpoint, how would that benefit you?
I think the diagnosis is important at every level, and the quicker we get it, the better it is. Obviously, from a point of treatment, we know that in most neurodegenerative diseases, the pathology in the brain is there, it's present way before the symptoms start. So what we know for Alzheimer's disease, usually people develop plaques and around 10, 15 years before they actually come to the memory clinic. And that obviously gives us an opportunity to intervene. And the earlier we intervene, the less damage may happen. Because once people get symptoms, the neurodegenerative disease process starts. The neurons start to die, the atrophy of the brain and the other markers start, and it's much more difficult to intervene at that stage. But also it's sort of optimizing environmental factors and environmental exposures. The earlier we know that something is going wrong, the better it may be in terms of influencing the outcome.
And I would imagine that would also make it easier to understand the disease, because from your definition of the longitudinal study is seeing people over time, but as we're humans studying humans, it's a hard statistical task because we all have the same lifespan.
It is, and especially for diseases that progress very slowly, like, for example, neurodegenerative diseases, people get worse over many years, and that makes things very challenging in terms of research, because you need to design a study that will be funded and will go on for many, many years, which is obviously much more difficult in that aspect.
Yeah, it's a hard task. I think in one of your interviews, I think on the page, the NHS page for the Essex Partnership University, you mentioned that other detection methods like lumbar puncture or nuclear medicine are not widely available. I don't think we have a lot of information of how these detection methods work.
Yeah.
But my question would be, why would you say they're not widely available? And are they as efficient as biomarker methods could be?
Yeah. I mean, at the moment, when it comes to Alzheimer's disease or other dementias, we give people what we call a clinical diagnosis. A person presents in the clinic, they present with a certain type of symptoms. We think they fit certain diagnostic criteria, and then based on the criteria that the person fits, we talk about the diagnosis of a probable condition. Without confirmation of the biomarker of that this is actually Alzheimer's disease pathology, research studies show actually that even the best clinicians, when it comes to diagnosing dementia, get it right only in about 80% of cases or 70% of cases. So someone might present fitting the clinical criteria of Alzheimer's disease, but they actually in the brain have the pathology of dementia with Lewy bodies. Mm-hmm. And that's why it's important to have the biomarker confirmation of the condition, so that we know someone who presents with Alzheimer's disease actually has the Alzheimer's pathology in their brain. And with biomarkers, we can do that. If we do a lumbar puncture, we can measure levels of amyloid beta and tau phosphorylation. So we are confident that actually the clinical presentation correlates with the brain pathology. And at the moment in clinical practice, we can either do that with a lumbar puncture or with a PET amyloid scan, where people have a radioligand sort of scan, a nuclear medicine scan, a PET scan that confirms specific presence of amyloid plaques in the brain. And obviously, talking about a lumbar puncture, you need to. It's like a day case. People need to come in the clinic. They need to be seen by a doctor. They need to have a lumbar puncture. They need to wait there for several hours afterwards. It's an interventional procedure. It's almost like a day case surgery. They need to stay lying flat afterwards, and the sample needs to be collected in a certain way, obviously, the spinal fluid, and then be sent to a lab. And there are only two labs in the country that do those tests, one in Manchester and one in London. So those are not widely available tests. It takes a long time. And again, similarly, an amyloid PET scan requires a nuclear medicine department, which is available in sort of big academic centers, but is not widely available for every patient. We think about how many people have dementia. The sheer numbers are huge, and the structure in place in the NHS are not sufficient to cover those diagnostic tests. That's why it's obviously important to develop and work on less invasive, more accessible tests. And blood tests are obviously widely available, can be collected very easily by any professional that is trained to do that, and then be sent to a lab that can be processed quickly and obviously at very low cost. A lumbar puncture or a PET amyloid scan may cost several thousand in terms of costs, healthcare costs. A blood sample, nowadays the cost is less than £100. So definitely it's much more widely available, cost effective, and possible to do in a large scale, which we require when it comes to diagnosing very common conditions like Alzheimer's disease.
Yes, definitely. And within the bounds of our project, we have identified the need for detection of Alzheimer's to be a screening process, so a standardized screening process that all citizens take upon a certain age, like bowel cancer detection by the NHS. And with a non-invasive method, that would be much more possible and I think much more beneficial for the public. But with this approach, we have heard several counter-arguments regarding the availability of treatments for this early stage. And what would you say the sort of current, not market, but situation of treatments for this earlier stage is? Can somebody who is diagnosed 10 years before be treated?
Certainly, there is the promise that prevention is perhaps more effective than treatment of active disease, and that's, I think, in a lot of diseases. The earlier we find evidence of something, the much more chances we have to treat it. And I think this is the case, at least with Alzheimer's disease, that we know the earlier we intervene, the better the outcome may be. So definitely there is the case for that. Having said that, I'm not sure whether we have enough evidence yet for a screening of the whole population. I think we still need to do more research as to what is the best marker to do that and how to best do that and if it's cost effective to do that. But yeah, I think that it's an exciting area of research, and people focus on that. There are all sorts of different ways. People focus on digital technologies as a means of screening the population, mobile phone use, cognitive tests, digital cognitive tests, other screening methods. But yeah, definitely there is a huge area to focus on.
Yeah, that's what we're trying to, where our research falls as well, because we're developing a diagnostic with biomarkers, but through the pathway of the gut, actually. Because we've read many studies mentioning the gut-brain axis, and we think that the gut is a good area to find biomarkers that could show that. And at this point, we are sort of at the point where we have to select the first biomarker to detect, and that's a hard task, because every biomarker could mean something different. I think in one of your studies, there is a database of biomarkers available. Can anybody have access to this database?
There are certainly lots of different, all of our research studies obviously are publicly available widely, and if something you cannot access, I'm very happy to share anything. That's quite common in the research community. But yeah, there are lots of different databases. The evidence obviously continues to develop and evolve. So yeah, definitely, those are great opportunities.
Now, because of course we are also new scientists, I would say, part of the audience of the podcast are people within the university or aspiring scientists. If you are willing to like to get into the path you took to end up researching this, was this always your choice, your interest to research this? Where did you start?
Yeah, I guess when I was at your stage, my personal journey, I was very interested in the brain. I knew I wanted to do neuroscience. I was interested in, understanding more about the brain and what goes wrong at brain conditions. And when I finished medical school, I wasn't sure what specialty I wanted to do. And actually, I went and I worked in a neuroscience lab. I did a PhD, which then it ended up being around aging of the brain and Alzheimer's disease. So that's sort of what took me to the path of becoming an old-age psychiatrist eventually, and combining research with clinical work. So yeah, I think research is a great opportunity to find what you're interested in, get involved, get in depth in a specific topic, and then build up your sort of interest and career around that in a sense. So yeah, it's great to do research, and obviously keeps you going in hard times of when the clinical work or things get very challenging.
Do you have any advice for students thinking about what they want to do with their studies or with their research?
Yeah, I guess do what you like, follow your interests, follow what you're interested in, because it's all about, passion, motivation, and enthusiasm. Those are the things that you need. And the other thing is keep going. There will be lots of failures. There will be lots of disappointment. Usually people who make it are the ones who are, managing failing well, because that's what happens most.
Yeah.
So yeah, it's keep going and follow your passion and your interest. That's the best advice.
So of course, to manage failure, you must be pretty interested in what you're doing. So what would you say is the most exciting discovery or work you have done recently?
Oh, I get excited every day. Mostly I get excited by things that don't get published or don't ever, show something. There are lots of exciting. I guess our work on epigenetics was quite exciting at the time. It was very novel. Actually, what I'm most interested about is a paper we have about to publish now. We used a method called laser microdissection, and we used that in post-mortem neurons of people with dementia with Lewy bodies, and we got neurons that have the pathology and neurons that don't have Lewy bodies from the same person. And then we screen their DNA and look for DNA methylation differences. And we find that even within the same brain, people who neurons have pathology of synuclein Lewy bodies are actually quite different when it comes to the epigenetic programming compared to neurons who don't have the pathology, even from the same person, which I think is quite exciting, and hopefully we will have a paper published soon.
Oh, we would really want to read that. So yeah, that falls within epigenetics. Can we, for the viewers, actually get a definition of epigenetics?
Yeah, it's again a difficult question. You asked me lots of difficult questions today. I guess where it all starts, when we talk about epigenetics, is what happens in addition to genetics, because all cells of a certain person or an organism have the same DNA. They have the same DNA sequence. But some cells end up becoming neurons, some cells end up becoming gut cells, some cells become blood cells, or all sorts of different deep types of cells. And the reason is because of epigenetic mechanisms, because of epigenetic processes. And there are lots of different ones. There is methylation of the DNA. There is obviously modifications in the histones that the DNA is wrapped around. And there is obviously several other ways that determine of how active or inactive a certain gene is. And obviously, by determining gene expression, a cell starts to sort of behave in a certain way, differentiate, in early development, but also function differently. So yeah, epigenetics are, I guess, the mechanisms that decide how a cell functions and what it becomes, or when something goes wrong, perhaps can tell us about why there is some problem there.
Talking about methylation, in one of your publications, you had mentioned methylation patterns. Could you go more in detail about how you identify these patterns?
Yeah, so there are all sorts of different ways to measure methylation. So methylation is a modification in one of the DNA bases in the cytosine of the DNA. As you know, we have 4 bases in the DNA that, create the sequence. And the cytosines can be modified. They can be methylated. They can be hydroxymethylated. There are even some other modifications. And depending on how methylated a certain region in the genome is, determines how open or closed the chromatin is, or how active that gene is. And we can measure that. You can isolate DNA, and then through various sort of biochemical techniques, you can actually measure the methylation of the DNA. And through sequencing those parts, you can tell that this gene is methylated in a certain area or is not methylated in a certain area, and that may tell you some information about how active that gene is at that particular tissues. And there are ways of focusing very deeply on one specific gene if you have a hypothesis, or there are ways of getting a snapshot using array methods that lots of different genes are measured at the same time, or sequencing, getting a piece of DNA and sequencing it to measure the methylation.
So, looking at this from like an engineering perspective, you're saying that, it can be identified and measured. Could that then be used as an effective diagnostic tool in any way?
It is used mostly in oncology research. It's used as a marker of oncology practice, as a marker of some tumors, of disease response, of subtyping. It is already used in certain areas that is more advanced. In neurodegenerative disease, there's still lots of research that we are doing, but it hasn't reached yet the level of evidence that we can use it as a diagnostic. But that's one of the things we are sort of looking at as well.
Do you feel like it could be a more efficient method of diagnosis should there be, like a correlation?
Definitely, because we know that DNA is very stable. You can get it from sort of every cell, but it's about, finding the right evidence to support that.
Right.
Technically, definitely, but it's all about, the evidence at the end of the day.
So you said that within the same brain, there were neurons that had the methylation patterns and neurons that didn't. So would that mean that maybe the disease causes the methylation rather than the methylation the disease?
Yeah, that's a very good question, whether what we see is actually a cause or a consequence of the disease. Mm-hmm. We haven't figured it out yet. We still try to figure out what's going on, but certainly there is lots to know whether actually the presence of the pathology affects how a cell behaves, affects the epigenome of the cell, or actually the epigenetic sort of is what started the pathology. It's still lots of research to do, lots of work to do to find out.
And so it sounds like a hard study to do because you sort of rely on donors. But how would you actually determine this? Can you monitor the genetics of a person that is still alive?
So yes and no.
Yeah.
Well, obviously we can get blood samples from people, and we're measuring methylation, DNA methylation in blood. But obviously, the DNA you get in blood is mostly from blood cells. Yeah. It may give us some information about immune changes that happen in a certain person, but it doesn't actually give us information as to what happens on their brain. Obviously, the genetic changes tend to be mostly tissue specific, or even brain area or brain cell type specific. So I guess that's where the post-mortem research is important.
And the method for dissection, I think you sounded pretty excited when you mentioned this. It was laser dissection.
Yeah.
So how does this work?
So that's, again, a method has been around for a while, but it's very challenging technically. So you get a piece of tissue, a brain slice that you put in the microscope. And then with certain immunohistochemical techniques, you can label neurons that have Lewy bodies and neurons that don't have Lewy bodies. And there are those microscopes that developed that the laser can actually go around. You can select, say, I want this cell, and the laser is so fine that will cut around one specific cell. And then the cell goes in a tube, and then you can select another neuron that looks healthy, and that goes in another tube. And that's, it's technology is, fascinating what sort of things you can do nowadays.
That's actually really cool.
It's really good, yeah.
Being able to identify cells and then just cutting them out.
And there are lots of different ways of, single cell sequencing. There are even automated ways that, single cells go through filters to be separated and do single cell sequencing. It's, the whole sort of technology is running very, very fast.
Yeah. This is really interesting, I think, to both of us.
Yeah.
Because we're actually, we're not regular medicine students, we're biomedical engineering students.
Yeah.
So I think...
Yeah, currently we're actually focused on microscopy and optics and understanding, how microscopes work. So the fact that this is a technique is actually really interesting to us.
Yeah, look it up. I mean, there are some very exciting videos online.
Oh, okay. That's very cool. We'll do that after the
podcast. How laser microdissection works. Yeah.
We'll mention it to our lecture.
Yeah, we'll definitely do.
This wasn't in the notes, but I just thought of a topic that we could go into. There's recently, I wouldn't say a new type, but a new focus in neurodegenerative diseases, which is CTE.
Is it chronic traumatic encephalopathy?
Yes.
CTE. Yes.
I think it's only recently been a focus. And it's speculated that it's caused by repeated head trauma, of course. Have you been involved or know anybody involved in research involving this?
Yeah, I'm actually involved in a study. It's called the Prevent Dementia Study.
Okay.
This study follows up people who have been. It's a multi-center study. It runs in Edinburgh and now St Andrews, in Dublin, in Cambridge, where I'm based, in Oxford, and in London. And there is a group of people that have been followed up for over 10 years now, that are fairly young in age, started when they were 40 to 58. Mm-hmm. And half of them had a family history of dementia, and the other half didn't have. And those people go through blood tests, brain scans, and they have lots of memory tests. And we sort of try to understand what factors sort of increase people's risk of getting dementia. And over the years, this study acquired a sort of second aim. It started recruiting people who were professional rugby players or professional football players to study exactly this effect. Because there is some evidence suggesting that people who are sort of exposed to repeated head injuries, for example, through sports or contact sports, are at increased risk of getting all sorts of neurological or neurodegenerative diseases. Mm-hmm. Chronic traumatic encephalopathy is one. People used to call it boxers' dementia because boxers, it was first described in people who were professional boxing players, but it seems to be the case in other professions. In football, for example, footballers are at increased risk of getting dementia, especially the previous generations that go to head balls that were very heavy, the first sort of earlier on. And now there are even advice that children who play football shouldn't hit the ball before a certain age. As a result of that, I think below the age of 10, something like that, there are guidelines saying that children are not allowed to hit the ball because exactly the risk of that. The risk is earlier on in life. But certainly, I do see in my clinic people who used to be professional football players, involved in wrestling, that are getting dementia. CTE has been described mostly recently. And people seem to get distinct pathologies. People seem to get tau tangles only. They don't get the full-blown Alzheimer's disease, but they seem to get the tangle pathology. That is obviously causing problems down the line. But yeah, it's still a very exciting area, and there is lots of research that is happening in the field that we try to understand and obviously inform the public as to if there are any ways to prevent that or what measures to take.
So are you part of the I think you mentioned that you're part of the Prevent Dementia project. Are you able to go more in detail as to what the project is about?
So the project is exactly what the title says. It follows up people and try to understand a little bit more what are the factors that increase the risk of someone that gets dementia, and if there are any ways that we can prevent it. It's more about, it's an observational study that goes on for several years, trying to understand more about what causes dementia and the mechanism behind that.
Yeah, and of course, with newly identified types of dementia like CP, we see that it can also affect the younger population, which is something that wasn't even considered, I think, with the rest of the types. And I think with all this new research, we continue to see that an overall awareness of someone's lifestyle and how not protective but preventative they are can help decrease their possibility of getting a neurodegenerative disease. In a previous episode, we talked about and we concluded that there should be a societal shift to prevent Alzheimer's, and we think that earlier detection could also help with this, because the earlier we can identify it, the earlier we can sort of try and prevent it. Would you say there are any significant actions to educate the population about how they can prevent neurodegenerative diseases?
Yeah, definitely. There is a lot of research out there, and it seems to obviously lots of different factors that improve cardiovascular health, also improve brain health. There is a lot of, obviously, the concept of brain health. There is the Lancet journal publishes the Lancet Commission on Dementia, looking at modifiable risk factors for dementia. It gets renewed every few years. I think there was one last year. I think it's certainly interesting. And that's so I think there were 11 or 12 modifiable risk factors that may account up to 40% of cases. And, things like improving hearing, correcting hearing, looking at cardiovascular health, education, preventing brain injuries, and all sorts of different things that we may be able to do. Reducing alcohol, obviously, smoking.
And as for the people who are diagnosed, are you also involved in the rehabilitation part?
Yeah, I mean, it's a huge societal, burden and cost, not only for the people who have dementia, but also for their families. Mm-hmm. And obviously people need a lot of care and people need a lot of support, and they do go through periods that, they may become more agitated, they may develop behavioral problems. And that's very difficult, and we don't have any good effective treatments for that aspect as well. And I think it's very important to talk about prevention and focusing on that. But there are many millions of people living with dementia at the moment, and we need to do something for them as well.
We've read that there are several approaches like cognitive exercises and in general an urgency of the families to stay involved with the individuals. Could you say what sort of significance regular cognitive exercise could have to
So there is good evidence that what we call cognitive stimulation therapy helps sort of to improve the outcomes. It's a structured therapy. We offer it in the clinic where I work. It's in the NHS, a program of what we call cognitive stimulation therapy. And people get to work with psychologists and occupational therapists in a group setting to go through various different activities that may help them, because in general, the evidence is the more physical and social activity that one has, the better it is for their brain outcomes. So keeping busy, meeting people, keeping the brain active and busy helps to sort of protect the neurons, help to slow down the neurodegeneration. And that's the general aim of those programs, to keep people engaged, keep doing activities. There is no specific, do a specific crossword or a specific brain training game that works. But in general, keeping the brain and the body busy, it's very good. And it sort of slows down the process.
I think that's what we're hoping from our screening test, is that with that given, years in advance, maybe some cognitive stimulation therapy or some environmental changes can occur where the patient doesn't have to go through such rigorous processes to treat themselves. They can, gradually get to that point.
Yeah.
But of course, a new possibility now is the disease-modifying drugs that are coming out. I'm sure you've heard of them, like donanemab and lecanemab. Were you involved in any research involving this?
I've not been directly involved, but yeah, they are treatments that are now approved by the MHRA, but they are not endorsed by NICE, so people can access them in the UK, but they're not on the NHS. There is evidence that they remove the amyloid pathology from the brain, and there are some improvement in the clinical outcomes, but the cost effectiveness there and the ease of giving them their infusions are not quite, to the point that the NHS supports them. The National Institute of Clinical Excellence does not think that they're of a cost. Their benefit compared to the cost and the risks that come with them are not there yet for wide-scale adoption.
So, do you have any knowledge of how this cost-effectiveness is evaluated?
Yeah, I mean, this is a very complex decision. I'm in no way qualified, to have. I can give you my personal opinion, but obviously, it's. I think there are multiple levels. Obviously, the efficacy is not massive yet. They are not, completely stopping the disease, or at least we don't have the evidence yet. So, the efficacy is not very large. They do come with significant risks. Those drugs, they can cause what people describe as amyloid-related imaging abnormalities, or brain swelling or brain bleeding in about 20% of people who get them. And they obviously come with huge costs in terms of, you need to have MRI scans to monitor for side effects, which are not widely available. You need to have infusion centers to receive them. So overall, the cost of such a treatment, compared to the efficacy that those have, that it's still not proven to be major, it made, I think, the committees to decide against adopting them in the NHS at this stage. This might happen later on, where there is more evidence about the longer-term benefits, or if there are new versions of them that are safer or more efficacious. I think they certainly open lots of new opportunities, because they do show that they have what we call disease-modifying properties, that there is a way to remove the pathology from the brain. And that is quite likely to be associated with better outcomes, but the research needs to evolve. And there are lots of new promising, newer drugs, longer period data that show that probably there is something that we will be using in the NHS as well in the near future. This is my personal opinion, obviously. The decisions are made by committees and experts in the field.
Yeah, of course. And it's really exciting that this field is evolving finally. This is modifying drug. It's quite an achievement if it can be effective. Would you think that earlier detection would make it even more cost effective to knowing, being able to accurately 100% diagnose who has it and who doesn't?
Definitely, and there are ongoing prevention trials as well. So those drugs are being tried in people who don't have the symptoms yet, as a means of preventing Alzheimer's disease. So,
I think that's quite almost all of our topics we've covered. So.
Yeah, it was a very exciting conversation, I have to say. It was really nice to talk to you, and we'd love to keep in touch with you.
I think for our listeners, if you could just let them know where they can read your publications, especially the new one that comes out, and if there's anything else that you want to mention to our viewers.
No, I mean, as I said, it's a very exciting field. What's the space? I mean, all of our publications are in peer-reviewed journals. I think Google Scholar is probably the easiest way to get the links to them. And yeah, I very much look forward to hearing more about your work as well. And yeah, I think it's an exciting field, and more things are going to happen in the near future. And what is most exciting, I think, how we look after people who have memory problems is changing. Has already changed since when I started practicing, and I think for you it's going to be a totally different game. I think neurodegenerative diseases will be treatable, and I think that's very good for the people who suffer and their families as well.
Yeah, we would love to see that happen. Yeah, thank you for this for your time in this interview. We love having a conversation with you. This has been George,
and this has been Bas,
and thank you to our listeners for listening to the podcast.